following is a presentation of Southern Evangelical Seminary. The purpose of SES is to train and prepare men and women based on the inerrant and infallible Word of God to evangelize the world and defend the historic Christian faith with courtesy and respect. To find out more, visit ses.edu.
My title is Director of Worldview Evangelism and Apologetics for Meekness and Truth Ministries, and if anybody would like to know more about that, we do have flyers here. I work with Dr. David Geisler, who wrote the book Conversational Evangelism. I also am a volunteer apologist with Reasons to Believe Science Apologetics Ministry, and so that's why we're doing this talk today. I took the class in microbiology with Dr. Fuzz Rana, and I've also done much reading and study and discussions over the years on these subjects. So I guess everyone knows the name of the subject, obviously. Does DNA equal God? Bioinformatics and intelligent design is the topic. Is anybody here in the medical field or scientific field? Okay, what do you guys do? Internist. Then you'll probably know a lot about the biological side. There we go. So when I talk about then you'll recognize how extremely simplified I've made the process of DNA expression as far as protein synthesis. I've left out error correction. I've left out all for simplification's sake. What we're going to do is we're going to take kind of a multidisciplinary approach in the sense that we're going to do the first part science on how it works, and then we're going to discuss a little bit about how recognizing the effects of minds versus random chance, just a little of that. And then finally, we're going to talk a little bit about how logic works. We'll cover a little bit of the science of it. We're going to cover a little bit of the pattern recognition, how minds work kind of thing, and we're going to cover a little bit on why the arguments a lot of people use from this information, why they're not as effective as they think they should be. And that's in other words, we're going to do a brief review of how some logic works. You're not going to learn everything about DNA today. I mean, obviously, you guys know that's forever. We're not discussing, as I said, we're not discussing epigenetics. We're not discussing error correction. We're doing a greatly simplified version, and yet in that in itself it's the gee whiz wow part of the presentation because it's just amazing what goes on in our cells. So, bioinformatics and intelligent design. What is DNA is the first part. We'll discuss exactly what it is. How is it expressed? That is, as far as proteins. Are there parallels to human minds? Worse and better ways to make a case for design. And you'll probably recognize a few of these things because many of us have read these arguments and we give them to our friends, and our friends who are not believers give us the dead-eye look sometimes, like so. And this will get a little bit why that works that way. Limitations of the arguments, which actually kind of blends in with the worst part. And then if we have time, we have a couple of DNA fun facts so that your brain doesn't completely explode if you're not used to this sort of information. And can the arguments persuade? And we'll see how that goes. Cellular composition. This is a eukaryotic cell. How do we know that? Because it has a nucleus. Prokaryotic cells are the more simple kinds. They don't have nucleus. The DNA in eukaryotic cells, like most plants and animals, resides in the nucleus primarily. And so this is the true nucleus, DNA contained in the nucleus of eukaryotic cells I mentioned. So what is DNA? There's where it is. Has anybody never seen the double helix DNA diagram before? I would be surprised these days if you hadn't at least seen it. It was discovered more than 50 years ago by Crick and Watson. You got the backbone here and on here, and then you got the nucleotides in the middle. The backbone's made of a sugar, deoxyribose, which is similar to ribose, which we'll get into in a moment. Just means deoxyribose just means they took a ribose sugar and they replaced an OH and an oxyl and replaced it with a hydrogen. It's just, anyway, not that important, and the phosphates. And then what's important is the bonds, these are covalently bonded, which means they're strongly bonded. They're sharing electrons. Those are strong bonds. So this part is very strong, whereas the center part, those are double and triple hydrogen bonds, which are very weak bonds. And you could think of it in terms of these are super glued together, and these are magnets holding each other together. And that becomes important because otherwise the cell isn't going to be able to do anything with the DNA. And I'd love to spend 2 or 3 days talking about problems and origins of life as far as naturalistic explanations because of things like this, but we can only briefly in an hour cover, or not even less than an hour, briefly cover a lot of these subjects. What is DNA? It's the most compact information storage system in existence. There's nothing we've ever invented as humans that comes close to the information storage capacity ability of what's in our cell, of what, according to the naturalist, evolved naturally. Chemical evolution would have been the initial way the information was stored, but either way, 1 teaspoon of DNA could hold the design information of all species of life that have ever existed and still have room to encode every book that has ever been written. That's in 1 teaspoon. Think about that. One computer scientist compared it, and he said that DNA is trillions of times more efficient at storage capacity than any chip we've ever invented. Does that kind of blow your mind? Is that amazing? So what is DNA? Technically deoxyribonucleic acid. It's a molecule that encodes the genetic instructions used in the development and functioning of all known living organisms. Everything has DNA that we know now. Now, once again, spinning back, there's a hypothesis called the RNA world that says the earliest molecules of life, the biomolecules, may have been RNA, but that's really... If you want to build RNA from scratch in like a prebiotic soup, the problem is RNA is orders of magnitude easier to break down than DNA. The bonds aren't as strong. It simply breaks down much easier. So getting it together doesn't solve the problems they're trying to solve by saying that RNA is a simpler molecule. It's not that much simpler, and then building it from parts up, it doesn't really solve that problem. Anyway, it is. So what's the key words for today's purposes? What's the key words we're talking about today that's in here? And that is, it encodes instructions. Now think about that. Think about the implications for a naturalistic versus a designed life kind of scenario. It doesn't matter whether you believe the Earth 6,000 years old or 4.6 billion years old. That's irrelevant to this argument. What's important is that it's a molecule that encodes instructions. As we talked about, the backbone is a sugar phosphate, and the base pairs bind the backbone together. Adenine always pairs with thymine. These are the nucleotides in the center that carry the actual information. And guanine binds with cytosine. As I mentioned, they are held together by hydrogen bonds, which are fairly easy to pull apart, as we'll see we're going to actually see an animation of this. I'm going to explain in detail, not complete detail, but in pretty good detail, how DNA is expressed in terms of protein. And then I'm going to show you an animation of a real-time animation of DNA being expressed as a protein. Once again, this is the greatly simplified view. There are wonderful books and years of study you could do on this subject. I think what's the key to understand as far as the information content, and this is key to me, what's really opened my eyes, is that the order these nucleotides are along the chain, along the double helix here, there's no chemical or physical necessity for them to be in any order. And if you think about it, that makes sense. In order to carry information, when you read a book, if the letters always had to be B, B, C, A, D, or T, H, E, T, H, E, T, H, E, if it had to, by necessity, the letters of an alphabet follow a certain order, it couldn't carry information. You would just have the same thing repeated again and again. But in order to carry information, it has to have the freedom to move the order of letters around in order to carry information. Does that make sense? Yeah, it does to me at least. And when I read a couple of books and first saw that, it really opened my eyes. That's why I got excited about talking about this subject this time. Well, we mentioned RNA. What's the difference between RNA and DNA? Well, I'm going to mention 4 differences here. As I briefly mentioned, RNA contains the sugar ribose. DNA contains deoxyribose. That's what you just take the ribose and you take out an OH, and replace it with just a hydrogen, and you get deoxyribose. If there's any chemist in here, I'm sure that makes perfect sense. For everybody else, you can just forget that. It's not that important. RNA contains the base uracil. DNA contains thymine instead. And that becomes important because we're going to get into transcription, which is going to be a description of how this happens, and you're going to see that in work. RNA is usually single-stranded, and not necessarily, but usually, and DNA is usually double-stranded. And RNA is short, 1 gene long at most as far as current life. Of course, we get back to hypothesis for early life, but those are hypothesis at this point. DNA is long, containing many genes. How is it expressed? How do our cells express DNA? What do they call that? Francis Crick called it the central dogma of molecular biology. That he meant this dogma in terms of super important. He didn't know that it means something you're supposed to accept without evidence on faith. He didn't really understand what the word dogma meant, but he really loved the religious language of it and what he thought it meant. And so he called it, later on he said he wished he hadn't used that term, but it once, like most things, once something becomes well known, it sticks. Okay, now we're going to get into how it's expressed. This is our cell, this rectangular shaped thing here, and to the best of my knowledge, perhaps in a plant, but I don't know any animals have cells that shape, but it fits the slide. There's our DNA, and this is, for example, this is going to be the gene we're going to express. Then it's transcribed, and what does transcribed mean in normal language? Well, copied. It's going to be change, scribe the writing. You're going to change the writing, so you're going to change from the 4 letters of DNA, which is, UTCG, you're going to change to, ATCG to AUCG. It's transcribed. They're changing one of the letters, and that's going to make it messenger RNA, which is then going to be translated, which means to change language, into a polypeptide chain via the transfer RNA molecules in the ribosome. We'll make it into a polypeptide, which becomes a collection of amino acids, which is a protein. Proteins can get to be quite large, as we'll see shortly. And there's a myriad problems with the origin of life. I can tell you that right now the origin of life studies they're going out in a zillion directions, and because to get from parts to completed machinery is a huge step. It's hard to emphasize just how complex any living thing is compared to non-living. It's really hard to emphasize. Protein synthesis, this is the same thing again, except we're looking at it in terms of flow of information. DNA to RNA to proteins. That's transcription and then translation. And transcription is a process by which a molecule of DNA is copied into a complementary strand of RNA. That is, you rewrite it. That's what transcribing is. You rewrite it, but you'll change one letter and then it'll be carried out. This is called messenger RNA. Once you get the complementary strand of RNA, that RNA it's going to copy is the messenger RNA, usually abbreviated as mRNA, because it acts as a messenger between the DNA and the ribosomes, which are in the cytoplasm outside the nucleus, where the protein synthesis is carried out. Once again, we're going to get into some more slides on how all this works, because we're going to really make sure we kind of get this, because this is so important as far as the design and information content argument involved here. Transcription process. RNA polymerase, an enzyme, attaches to DNA at a special sequence that serves as a start signal, and we're not going to get into all that detail, but basically it's going to search until it finds where it's supposed to start copying the information it needs to make the protein. And the DNA strands will be separated. As I mentioned, the bonds between the nucleotides are basically weaker bonds and easier to pull apart, so they're going to pull it apart so you can separate it. And then one strand will serve as a template. Now, since A and T always go together and C and G always goes together, whether you go one way or the other way, 5 to 3 as they call it, either way you go, it's going to be the same information. It's just a matter of which way it goes. And the RNA base attaches to the complementary DNA template, thus synthesizing messenger RNA at that point. And we're going to look at some more diagrams of that. We're going to look at a video of it, which is my favorite part, actually. Transcription process. The RNA polymerase recognizes a termination site on the DNA molecule and then lets go. It releases the new messenger RNA molecule. And remember that in textbooks, they'll say things like, gives the appearance of design. Or one textbook that says, actually has in parentheses, you must remind yourself that this evolved and was not designed. Now, why would you put that in there? Because you believe it was evolved, not because you can prove it. It's an assumption, and you want to make sure all your students know exactly what you believe.
Wasn't that Richard Dawkins that said that?
Yes, it was indeed.
Yeah, which is amazing.
Yeah. Oh, exactly. What he's saying is, I see design here, but I don't want you to see design here. That's what he's saying. Messenger RNA leaves the nucleus through pores and travels to the ribosome and the cytoplasm, which is the goo outside of the nucleus inside the cell. Was that too technical? The goo outside the nucleus, was that okay? You liked it? Yeah, I understand. I fully realize we spent many hours studying this stuff, just how much information it can be. And I'm trying to, where I can, simplify and just make it general. As I mentioned, we're not going to discuss error correction. We're not going to discuss splicing. We're not going to discuss epigenetics. These are areas of levels of difficulty, actually, for the materialist viewpoint, to explain that a person who believes there's a mind behind it has much easier time explaining. And why do I think this is so important? It's because what you see in physics, I mean, in the 60s, after the Big Bang Theory became pretty well established that the universe did begin scientifically, one physicist complained that every physicist he knew was running off to join the first church of Christ of the Big Bang. In other words, the theological implications of an origin of the universe was so powerful that the guys were starting to see something beyond that. And what's happened is in biology, because of Darwin's theory of evolution, material causes and natural causes, natural selection and such, the biologists have been lagging behind the physicists in recognizing the need for God. And as he mentioned, Richard Dawkins, who's written The God Delusion and The Selfish Gene and other books, just to combination to try to sell blind force evolutionary theory, and number two, to ridicule belief, particularly Christianity. He says he's talking about Islam and Christianity and all the religions in general, but typically he's terrified of Islamic terrorists, so he rarely mentions Islam. He does have self-preservation. Okay, this is the part where the polymerase is, you see here, this is the DNA. It's here it's still joined, here it's been split, and here is the RNA coming off, and the polymerase is going to make that RNA copying the nucleotides on this side, and they're colored. So adenine, guanine, cytosine, and thymine on the DNA here becomes adenine, guanine, cytosine, and uracil on the RNA side. So it's transcribing it. Transcription is changing one letter in order to carry it out. Eukaryotic, once again, eukaryotic means true nucleus type cell. Eukaryotic transcription, we're going to have the DNA there, and then it's going to be transcribed to RNA. And here's our nucleus again. And the RNA is going to process, it's going to need to be processed, so we get messenger RNA. And then it's going to, those are nuclear pores, little holes in the nucleus that allow it to go. It's going to be exported into the cytoplasm. Did everybody get all that? This is like the third time, and I only, like I said, I'm only repeating it because this is the key part of the thing as far as the physics of it goes, as far as how it chemically works. This is the key part here. You know, I can tell you that, to talk about the complexity of cells, I know a guy who said he knew another fellow who studied, and he got his PhD because he did his PhD research on pores. Because pores are so complex, the study of those are worthy of getting a PhD. That's how complex just one part of the cell is. It's really mind-blowing when you get into the complexity. Translation, that's the second part. Transcription, first part, translation, second part, is the process of decoding a messenger RNA molecule into a polypeptide chain. And poly just means many, peptides, which are the amino acids chain, which become a protein. Just check and see what time we're supposed to be done so I don't run too far. Okay, good, we got a half hour. We're going to have time for questions and fun facts. Each combination of three nucleotides, those are those little base pairs along the line on messenger RNA, is called a codon, or a three-letter word. You see why they're using the words like transcription and translation, language words? It's because we're talking about, and these aren't coming from Christian books. These are coming from standard explanations of how DNA works. Each codon specifies a particular amino acid. So each three-letter word along the line of the chain of the mRNA specifies a particular amino acid that is to be placed in the polypeptide chain protein. So what happens if it puts it in the wrong order? The cell will recognize it's not right and break it down and start over again. But that just evolved that way. There's no design in it. I mean, anybody who ever worked in a factory can look at a cell, or ever run a computer can look at a cell, and it screams, this is a result of a mind. And I can tell you that it's either Crick or Watson, one of the two guys who discovered the double helix nature of DNA, saw the complexity of life. And so he said it had to be put here. And so he came up with a theory called directed panspermia. Has anybody heard of that one? Aliens, exactly right. Directed meaning purposely, panspermia meaning spread out the seed of life. Aliens put life here because he's a scientist. And scientists don't believe in God, he knows that. But certainly aliens that's respectable. God, no, we can't talk about that. Translation part. Those are the codons. We talk about the three-letter words, AUG, AAC. They see the orders vary according to what amino acid is trying to tell it to build, to add on. A three-letter code is used because there are 20 different amino acids that are used to make proteins. There's about 100 amino acids available, but life typically uses about 20 of them, not all of them. So the first life, if there were other amino acids available, it said, nah, we don't need you. If a two-letter code instead of a three-letter code were used, it would not be enough codons to select for all 20 amino acids. So life basically requires that three-letter code, that 3 letters. It works like this. That is, there are 4 base pairs in RNA, so 4 squared, which is 4 times 4, is 16. 16 is not 20 amino acids you need, whereas 4 times 3 is 4 to the cubed. 4 times 4 times 4 is 64, so that certainly gives you enough information to code for 20 amino acids. There's actually some redundancy. The amino acids, some proteins don't require an exact, there are certain amino acids that can replace others and not affect the function. But typically, and some of you guys in here will know this, when it comes to the folding, the 4 levels of the protein, when it comes to the folding and such, typically it's critical that they all follow the exact correct order, which is why cells have other mechanisms built in to correct, either error correct or destroy the protein when it's not done right. Because we have a finite amount of energy we can take in through food, and your cells have to work as efficiently as possible so we can stay upright. Okay, here we go. We're going to see if this is going to work this time. Yay, it looks like it's going to. Let me give you a quick, all right, there's your DNA, double helix. Oops, that didn't work at all. We're going to go back and try that again. Okay, we're going to see the DNA. We're going to see the polymerase come in. We're going to see it, and once it gets the collection of molecules it needs to build what it needs efficiently, we're going to see it zip down the DNA, and we're going to see the RNA coming off the side. Then we're going to see the RNA go off, and then we're going to see the messenger RNA go out. We're going to see it go out. We're going to see the ribosome grab it, the 2 parts of the ribosome grab it, and we're going to see the manufacturing of a protein. And then we're going to discuss just how bizarre that is. But here it goes.
The innermost workings of how a simple code is turned into flesh and blood. This is what Francis Crick called the central dogma of modern biology: how DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left. Everything's ready to roll. 3, 2, 1. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, It snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. They all carry a specific 3-letter code that will be read by the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecule. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second.
So if you go back to the origin of life, and the earliest signs of life as far as not carbon dating, but radiometric dating and such, the earliest signs of life, they date back to about 3.85 billion years ago at the earliest. And if anybody knows their astronomy and such, up to that point, up to about 100 million years before that, was the late Hadean bombardment, where they say when the Earth was being formed, it was being slammed by huge asteroids and meteorites and such. And so the Earth's surface was, they hypothesized, was molten to about 200 kilometers down. And as soon as it cooled enough that there could be liquid water, there's evidence of life on the early Earth. And not just early life, simple life either. We're talking about the blue-green algae, cyanobacteria, which uses sunlight to process. If you study plants and such, processing energy from the sun is not as simple. And so I've done the study from the secular side, and so what one of the current leading hypotheses for how life could have begun, how cells could have begun, is that it used chemical energy and happened within rocks and such. That certain molecules of that have certain chemical energy and such, and that they're trying to find ways for this to happen because processing sunlight energy is a very complex. Photosynthesis is very complex, but that's the earliest life we can find. So we say, well, where's your physical evidence for your earlier life? Do you have any evidence for anything that wasn't complex already? And they say, well, here's what we think happened. All the earliest life we have evidence ate all the earlier evidence. I mean, I'm not making this up. Literally, they say the evidence isn't there because it was eaten by the earliest life we have evidence of, which was really complex. But the simple stuff that formed naturally, that got all eaten up. All of it. Every bit. It could have been aliens planting life-eating things. You can indeed. Yeah, you talk about hot water vents and such. I'm sure you know the problems with that. Is that proteins, amino acid chains and such, those kind of chemical bonds break really easy according to when the water gets warmer especially. And they're all water soluble. And so theoretically, yes, you get energy from the vents and such, and if you have a compress, certain reactions work differently under high pressure and high temperature, and so they're hoping that they can find a way for that to work. So that is one of the current models they're looking at right now. Do you have any more information about that you'd like to share?
That same event happens in the body of the ocean. There's no life.
Right. Oh, exactly. You're talking about extremophiles right now. Yeah, sure, absolutely. You guys are familiar with the term extremophiles? It just means life that's in an extreme environment. Phile means, like philos in Greek, love. They love extreme conditions. So there's life that exists within very cold, very high pressure, and also life that exists in the like the underwater vents. Incredibly complex life. But it's much easier for life to adapt to that environment than it is for life to begin in that environment. I mean, if you get to the whole, I mean, this is not from a Christian perspective, but Stanley Miller and Urey, the guys that did the whole origin of life when they synthesized a few amino acids in the test tube, University of Chicago, 1950s, those guys hate that theory. Well, Urey's dead, but Miller and his disciples hate that theory. They mock it. They say, why are you even studying something that couldn't happen to something that life can adapt, but it could not start there? And that's their opinion on that. But you do. I mean, that's why I said there's a billion directions this origin of life research is going, because they haven't found any good single way yet, and so they're trying some very interesting and they're discovering some interesting stuff. But as far as how to get an entire living cell, I mean, you actually have three different camps. You got the cell membrane camp. That the first thing that chemically evolved was cell membranes, because you have to have an enclosure to let the good stuff in and the bad stuff out. So they had to evolve first, they say. And the other people say, no, you have to have metabolism. You have to be able to process energy first, because it's not alive if it doesn't process energy. So that came first, and then a cell membrane. And another group says, no. You have to have replication, or else the first thing lives and then dies and it's gone. So so other people say, wait. You have to have all three of those in the first thing. And they go, well, don't bother us. We're trying to break it down into parts we can test and discuss. But it's as you know, if you obviously studied it, it's incredibly complex, and they're going in some very interesting directions. I'm not very confident that professor at Duke University of Biology, he said, not only do we not know how life began, I don't believe we'll ever know. And he's not a religious person at all. I guarantee that. But, you know. Do I think virus is alive? Not personally, but there's some people who do. So they can't live. They have to hijack DNA from other things. So does that count as life? I don't think so. What do you think?
No.
Yeah. Good. That means. Yeah.
I have to think of all possibilities.
Absolutely. But we're going to get into why this is not conclusive, and why bringing this to a person who doesn't believe is not going to persuade them in and of itself. This kind of information to us, we look at it and we say, it screams God. Why don't you see that? I mean, does everybody not have that feeling when they see this? Yeah, and that's why I talk about the third part of the discussion, is why it's not persuasive as we want it to be. Okay, so we talked about the parallels here. Language of creation, we call it. What does language have? Language has an encoding system, such as an alphabet. It doesn't have to have an alphabet, but it does have an encoding system as far as our experience. Most languages ever spoken don't have an alphabet, but we know that languages have an alphabet now, ours. It has a grammar, has meaning, it has intent. That's what language does. What constitutes language? Those things. Why DNA has those same things. That's why the talk's called, Does DNA Equal God? Does DNA demonstrate that God must exist? How about this? Can anybody read that? What happens if you take our alphabet and convert it to DNA, to the nucleotides?
Oh my goodness gracious!
Now, does that not help it stand out that it's now, I don't know that this particular sequence exists. I put this into a translator in order to turn words into DNA language. But the point is, our language can be converted to DNA because it's an information-bearing molecule. It carries information. Where does information come from? Matter and energy all by themselves cannot produce information. They can serve as medium for storing or transporting information. And there's two different kinds of information. We're not going to get into all the philosophical details, but basically there's random information, such as say you cut up 100,000 letters of the alphabet, cut them up, drop them in a hat, shake them up. Well, that's information. Whatever order it comes out, that is information. But it's not usable, it's not practical, it's not meaningful information. That thing I just showed you, and there it is. There's my little pointer thingy. Can you see that? All those letters, that's usable, meaningful information. So, if I hold up, can anybody read the cover? It says the cell's design, doesn't it? If you were to look at this as an artifact, Well, not even an artifact, because that comes from people. But if you look at this as a natural object, if you're a scientist, you would study this and you could tell us the chemical composition of the paper. You could tell us what kind of inks and how they reflect, what angstroms of light, etc., they represent. But you would be missing something there. There's something besides the height, weight, width, density, etc. here. There's information here that goes beyond the physical. Physical medium carries information, but it does not produce the information in our experience. And once we're going to get into how not to use that, because that sounds like I'm saying something I don't want to say, so we'll get into that. Information cannot be created without intent. That is, we have no examples of information being created without content. Does that make sense? There are no examples of information that are created without intent. No examples I can think of. Possible objections to these types of arguments. But you have 98% of your DNA in common with a chimpanzee. You hear that all the time, don't you? Have you guys heard that? Yeah. Does anybody know what the real number is? Actually, right around 90%. And does that make sense? Does it not make sense that the DNA to make a finger, the instructions to make a finger, would be the same whether it was a monkey or a human finger? Does that not make sense? Is that surprising? Well, here's some possible responses you might want to give. Yes, we're 90% in common with cats. We have 80% in common with cows. 75% in common with mice. Fruit flies, 60% DNA in common. Bananas, 50%. Humans and cabbage share about 40 to 50% human common DNA. That proves conclusively we evolved from cabbage. Can anybody possibly excuse? No, I'm just. So my question is, does this look to you like an example of common descent or like a common plan? That is, that the creator using the same medium to carry information in a common instruction plan or common descent. A materialist would argue it's common descent, and a Christian would argue in some sense that it's a result of a mind. What about that 98% of junk DNA? Have you heard that one? 98% of our DNA is junk DNA? Yeah, that's a common one. The word junk DNA. And now we're going to get to why that's wrong, but this is a new project started a couple of years. The Encyclopedia of DNA Elements, known as ENCODE project, suggested in September 2012 that over 80% of DNA in the human genome serves some purpose, biochemically speaking. So that 98% of your DNA is, excuse me? Somebody say, 98% of your DNA being junk is just false. It is an atheism of the gaps. It is saying we don't know what this other DNA does. We know this part, it codes for proteins and such. But we don't know what this other part does, therefore it does nothing. It's junk. Do you not see how that's an atheism of the gaps type argument? We don't know, therefore it must be junk. Well, they're starting to discover epigenetics and other stuff that I'm sure our special guest in the back would be able to explain better than I, but we're starting to understand that there's a lot more going on there. I think it's hubris, it's arrogance to say we don't understand it and we must know so much it must be junk. That's just nonsense. How far has science advanced in saying, okay, we know all about this, and then 20 years later go, oh, wait a minute, we were wrong about that. There's a lot more going on than we thought. Well, they're discovering that. So that's an atheism of the gaps. It's kind of like the God of the gaps argument. Anybody familiar with the term God of the gaps? Yeah. You don't know how it happened, therefore God did it. These arguments are discredited and outmoded approach to apologetics in which a gap in scientific knowledge is used as evidence for the existence of God. You know, they didn't used to know how carbons were synthesized for there to be enough. You know, the earliest universe, after the Big Bang, according to standard scientific models, about 80% hydrogen and about 20% helium, or a couple percent of lithium, but none of the heavier elements. And so, where do the different elements come from, and how do we have so much carbon on Earth? Those are the questions they couldn't answer before. And so, some helpful Christians at the time said, well, God did it. You don't know, so God did it. And this has happened several times, and it's an embarrassment. It becomes an embarrassment. And that is, you say, you don't know, therefore God did it. Well, see, the problem is, that's not the kind of God, that's not the God of the Bible. The God of the Bible doesn't step in. That's the God of deism at best, where He created the universe, wound it up, and let it go. That's not the God of the Bible. From a philosophical point of view, the inherent problem of the God of the gaps apologetic is that it relegates God to only a point of creation, the point that we do not yet understand. They say, this happened naturally without God's help, but He stepped in here, that kind of idea. The problem of the God of the gaps apologetic is that the gaps are getting smaller. We are learning. Science is progressing. It's a wonderful thing. I love science, and I dreamed of going into science when I was a young man, but young men do silly things sometimes. You end up in other areas. God of the gaps argument is based on what we don't know, and this is why what I'm talking about today is not a God of the gaps argument. That's what I'm trying to emphasize here. The design inference, that is the inference that there's a designer to life, is based on what we do know. And naturalism, when it is saying, is employing a no God of the gaps type argument. What they're saying is, in the future, we'll know how all this happens. Well, that's a utopianism, but it's not a logical or a compelling argument. What's the biggest problem with the God of the gaps arguments where we say, well, you don't know, therefore God did it, is that it's an informal logical fallacy known as argument from silence. You can't prove me wrong, so I must be right. Well, how about this? President Nixon built this building. You have to accept that because you can't prove me wrong. See, that's the argument from silence. It doesn't prove anything. All it proves is that you can't prove I'm wrong. It doesn't prove that I'm right. This says that because you can't prove me wrong, I must be right. Wow, how did I know that was coming? Okay, I meant promise, and we only got a few minutes here, that I'd end up with a few ways of reasoning, that is, ways that we can reason better or worse. Deductive reasoning. Are we all familiar with deductive reasoning? That is, you link premises with conclusions. If all the premises are true, and the terms are clear, and the rules of deductive logic are followed, then the conclusions reached are necessarily true. All men are mortal. Socrates is a man. Therefore, Socrates is mortal. Is that a percentage answer? Is there an 80% chance right? No, it's a necessarily true statement, deductive logic. That's how it works. Is what we're talking about with this design inference, is that a deductive argument? No, it doesn't, and that's what we want it to be, but it's not. It doesn't prove it in that form. Is it inductive reasoning, which is what science is based on? This is reasoning which the premises seek to supply strong evidence for, but not absolute proof of the truth of a conclusion. The truth of an inductive argument is supposed to be probable based on the evidence given. Scientific reasoning works this way. Let's say scientists, naturalists go out looking and they find every swan they've ever discovered is white. And so they put in the books, swans are white. And then one day they discover a blue swan or a green swan or something. Well, then they go back and they say, we were wrong. The inductive arguments don't prove it, but it gives a percentage based on how much evidence you have as how strong the argument is. Are we using that sort of argument? No, I didn't give you a percentage. We are using abductive reasoning, and this is not one that's commonly heard of. It's a form of logical inference that goes from observation to a hypothesis that accounts for the reliable data that is the observation, and seeks to explain relevant evidence, also known as argument to best explanation. Let's say you walk into a room and there's a big jar of jelly beans there, and there's jelly beans on the table. Could you prove deductively that those jelly beans came from the jar? No. Could you prove inductively? No. It's abductive reasoning, but you're certainly reasonable to infer that the jelly beans came from the jar. Can you see that this is a level of proof that's below the other two? Like one is absolute, one is probability based, and one is just this is the kind of argument I'm using and that we use when you talk about design. If you see it, you see it, but you can't use it to prove God's existence. That's what I'm saying. There's limitations to this. And once again, I forgot what time I'm supposed to be done, so oh, there we go. We got another 4 minutes. How do we use it? With humility. We don't go in there and we don't pound them and say, here's this information, bam, there's God. We don't do that. It doesn't work. We're using analogous reasoning. We're saying analogy. It's a form of argument to best explanation, abductive reasoning. That's what we're using. It's a lower form of proof. It's certainly, we do abductive reasoning all the time. We use it all the time. It's generally reliable, but it does not have the level of force of the others. But by using an analogous reasoning, we are not doing a God of the gaps argument. You see the difference here. We're not saying God must have created this stuff, but we're saying every example of information, encoded information we know of, is from humans or from minds. Therefore, we're reasonable to believe that God created life. I would like to say that this stuff was absolute slam dunk proof, but as anyone who's ever tried to use this sort of apologetic with non-believers knows, it's helpful. Well, how do we use it? Part of it's our attitude. We don't go there and pound them and say, you have to believe in God because of this, because that simply doesn't work. We say, here's what I see, and can you see why I believe that the informational content of DNA is more similar to what we know comes from minds instead of blind forces? That is, we're trying to make them an ally, make us common people who don't know everything. We're not speaking from on top of the mount, and let them understand this. Can you see? And try to get them to look with us. We're not talking down to them. We're getting them to try to look together with us and try to understand us a little bit more. I mean, we talked about the piece of paper. I showed you the book where there's information on it. You can analyze it chemically and physically, but with the missing element on that, well, the information. How about a CD? A CD with the Library of Congress on it and a blank CD. What's the difference if you measure the weight, etc., size? No difference. What's the missing element? Information. You can say to the person, do you see that matter and energy may carry information, but is not a likely source for the origin of that information? Notice they're not a likely source. Can you kind of see where we're coming from on this? It's not proof, but it's part of the support for a total overall apologetic or argument for God. Immanuel Kant noticed, he said, and this is not a quote from him, obviously, we can reason from intelligent design to a mind, but not to the God of the Bible directly. There's just so much more involved. We cannot say that this shows a mind, therefore God of the Bible exists. We see the step, but you got a lot more steps to go. Anybody who's tried to use these kind of arguments knows that. Anthony Flew. Did anybody know Anthony Flew? He was the world's foremost philosophical atheist. For more than 50 years, he wrote books on supporting atheism from a philosophical perspective. He was the go-to guy for atheists for more than 50 years. In a 2004 symposium, he announced his new belief of the world. His change of heart, he said, was almost entirely because of the DNA investigations. This is the man who was for 50 years, and you see the title, There Is No God, No Crossed Out, There Is a God, How the World's Most Notorious Atheist Changed His Mind. Now, did DNA, he called it almost entirely, but you notice he didn't say entirely. He began to study, actually it was Aristotle's philosophy that helped him to move to the idea of a prime mover and such to get the universe going, things that he had not thought of until he really went back and studied Aristotle again. By the way, Southern Evangelical Seminary loves Aristotle, so and then you see this guy, you can kind of understand why they love Aristotle, but almost entirely because of DNA investigation. So, DNA, the mind behind it, the design, that's just amazing stuff, and that's what helped to convince him that there must be a God. Is he a Christian? No, he's not a Christian, but he believes in a creator and a designer now, so that's where it takes you. Does it take you to the God of the Bible? Didn't take him, doesn't take most people, but it's helpful. A couple of fun facts. If you could type 60 words a minute, 8 hours a day, it would take you about 50 years to type the human genome. Think about that. Besides being really tired and bored. This is my favorite one. Each cell contains roughly 2 meters, about 2 yards of DNA. You have about 100 trillion cells in your body. Therefore, if you put all the DNA molecules in your body end to end, the DNA would reach from the Earth to the Sun and back over 600 times. See, I mean, here's a new thing. If you got, they have recently stored an entire genetics textbook in less than a picogram of DNA. That's 1/1,000,000th of a gram. They've actually encoded it into DNA. Now think about this as far as data storage and stuff. I mean, they've discovered DNA that they date back 20,000 years. So think about any CD or cassette tape. How long do those last? Yeah. And here's too much data for one hard disk. Put it on DNA. Tom Ryan says, this is a computer scientist from Israel. This is the final slide. He says we can get 3 trillion computers working in parallel in a space the size of a water droplet. The O's and 1's of conventional computers are replaced with the 4 DNA bases, A, C, G, and T. Operations can be translated into strands of DNA using these bases, and the way the DNA strands interact with each other produce new strands which can be decoded as output value. That is, this is a theoretical computer construct, a DNA computer. Think about it, drop of water, trillions of computers in parallel. And don't tell the NSA. Is this from chance and necessity? I have trouble believing that. What did Bill Gates say, finally? DNA is like a computer program, but far, far more advanced than any software ever created. If anybody's ever watched their Windows give you the blue screen of death, you know that they need to be a lot more creative. And the morality of science, they want to disassociate morality from science. Science is about facts, morality is from religion and such. There's tons of problems that can come from this kind of information. And that's why we need people around like us standing in the gap.
Amen.
All right, thank you all for coming today. Thank you.